share a new one, which will be a very common topic that you should be very happy to work on it and know about it. So where's this? So we share the screen and we go to, okay, we talk about glycolysis. Now we are stepping into biochemistry part of it, a chemistry part of it, where a lot of reactions and structures will be involved, and this will be building upon what information you already have. So when we look at glycolysis, we look at it as a standard procedure in all bacteria, but then there are variations in different bacteria that we will emphasize. So I need to know that you understand what glycolysis is all about, and you remember the glycolytic pathway. Then only I can define some of the variations in different bacteria. How many of you know what is glycolysis? Anybody understands what the word glycolysis and the pathway which we call glycolysis is? Yes? Breakdown of glucose to make what? A 3-carbon compound? Pyruvate. So glycolysis goes from glucose oxidation to pyruvate. This is an oxidative process where glucose, a 6-carbon compound, will be converted to pyruvate, which is a 3-carbon compound, and then further breakdown of the pyruvate will occur. So what is the purpose of glucose oxidation, first of all? Why does the cell wants to break down the 6-carbon compound and bring it down to the 3-carbon compound? What is happening during glycolysis? It is making certain products that will be useful for other processes. Other than that, what could be the purpose of any oxidation? Make ATP? Yeah, to produce energy for the cell, right? So this is an energy producing pathway that we will talk about, right? So if you look at the summary of glycolysis, this is the summary, right? Glucose using up to ADPs, adenosine diphosphate, and 2 inorganic phosphates, and 2 NAD. We'll talk about what is NAD. And then it will go into 2 pyruvates, 2 3-carbon compounds. It will release 2 ATPs, and it will have donated some electrons to NAD, which is an electron carrier, and convert it from NAD to NADH plus H, right? This is how we write it. So remember, what are we getting out of glycolysis? 2 3-carbon compounds, 2 ATPs total, and some carriers that are now carrying the electrons that were taken or ripped away from the glucose, and they will be carrying it to the electron transport chain and be able to produce more energy for the cell, right? So the whole load down, you understand the summary of glycolysis. So let's look at it as such. Oh, we've gone to the last one, so let's go ahead, go back to start with, review. So some of the terms that you should know and you should work on it over the weekend or before the next class is what is metabolism, what is catabolism, anabolism, enzymes which are called holoenzymes, apoenzymes, cofactors, coenzymes, prosthetic groups, role of ATP, role of oxidation reduction in metabolism, electron carriers such as NAD, NADP. So I will go through some of these very quickly because I expect you to know. So these are reviews that we will run through as much as we can right now. Okay. So what is common aspect of metabolism that is common in all organisms, whether they are bacteria or anything? So we have the laws of thermodynamics. That means energy must be obtained from the environment, which would be nutrients. That means energy cannot be created until you feed the bacteria or the cell with nutrients, right? Energy obtained in most are often conserved as ATP. This is the short-term conservation of energy, which is ATP. Not only is ATP the source of driving some of these reactions in the cell, but oxidation reduction reactions also play a critical role in energy conservation. And many of the oxidation reduction reactions occur in the electron transport chain, and we'll talk about the electron transport chain very soon. Chemical reactions that occur in the cell are organized in pathways. You're familiar with it, that they have to be in a pathway which allows the maximum efficiency in the cell. So the product of one reaction acts as a substrate for the next reaction and so on. So they are organized in these pathways, as we call them. Now the reactions are catalyzed by enzymes, and now we also know that there are certain enzymes that are considered as ribozymes. That means they are made up of RNA, but they have enzymatic activity, and so you will come across enzymes and riboenzymes terms, so watch out for that. And of course, all these biochemical pathways that we create and we looked at and we've studied have to be regulated so that they are not going forward, backward, and wasting all the energy. So this regulation of these pathways is going to be a major topic for us as we go along in bacterial physiology. So we are interested in the reactions, but we are also interested in the regulation of these pathways. So let's look at what is the major nutritional types of microorganisms, and we've discussed this before, that based on their carbon source, on their energy source, and the electron source, they have their different names. So we have photolithoautotrophs that use carbon dioxide as their carbon source, and then light as their energy source, and inorganic electron donors. And these will be a special group of bacteria that we'll come up with now. Purple and green sulfur bacteria, cyanobacteria, and diatoms belong to this category, and so on. Up to now, you have only studied this last group. So what I'm going to expand your information in the energy production by all these new different types of bacteria now. Okay, so you have only done this one. We need to move on to the others, is my idea now. Okay, so we go ahead and talk a little bit about fueling reaction. Even though there is a huge diversity in the energy, electron, and carbon sources that are used by organisms, they all have the same basic needs. They should have ATP as its energy currency. It should have some reducing power to supply electrons for chemical reactions, and they should have precursor metabolites for biosynthesis. So these smaller precursor metabolites are something that we will focus on in a little bit, and then we'll talk about the energy ATP and your electrons that will be supplied by the oxidation reduction reaction. Okay, so where are these channeled into? So if you look at it, your precursor metabolites are coming from your carbon source, which your autotrophs will use carbon dioxide as their carbon source, whilst the heterotrophs will use organic molecules as their carbon source. And from these carbon sources, we will produce some precursor metabolites. We will focus on this word in a little bit. Your energy sources will supply the ATP. So in your chemoorganotrophs, your organic molecules will provide the ATP. In the chemolithotrophs, inorganic molecules will supply the ATP. In the phototrophs, light will provide the ATP. Whilst your electron sources will be provided in the organotrophs by the organic molecules, and your electron source in the lithotrophs will be provided by inorganic molecules. So this reducing power will be applied, and the ATP will be applied also, either to make the precursors or move the precursors into the monomeric or the building blocks. And eventually the building blocks will be converted into macromolecules with the use of ATP. And finally, your pretty little bacterium will be all completely synthesized. So all this fueling reactions is what we're going to be studying, as to where the ATP fits in, where the reducing power fits in, what are the precursor metabolites that will eventually make your monomers for your proteins, such as your amino acids for the proteins, such as your monosaccharides for your polysaccharides, and your ribonucleic acids, where we will be using the monomers also, and make those macromolecules, and eventually make the 4 macromolecules, which we have lipids, we have your proteins, we have your sugars, and we have your nucleic acids. these 4 macromolecule synthesis is what we will look at, which will eventually make your bacterial cell. Are you with me to understand what we are discussing and how we can put together this idea that we are actually making a bacterium now, and we will definitely be using these ideas as to how does the cell make all these components that will fit into the structures of the different structures in the bacteria. Now, in microorganisms, when we talk about bacteria, some have great metabolic flexibility based on the environmental, what the environment is. So sometimes you will see that they will move from one nutritional type to another nutritional type if they are not provided with the correct one. So there is flexibility in bacteria in terms of their metabolic capabilities, and definitely that does occur in bacteria more easily than in eukarya itself. Since these bacteria are subjected to frequent environmental changes, they will have to adapt to those environments and be able to survive using whatever is being given to them. Right? So cells must do work, we know that. And what is this work that we are talking about? What kind of work do? They have to do chemical work where they synthesize complex molecules. Each cell has to use transport systems for transport work, where nutrients are to be taken in, waste products have to be eliminated, and also to maintain ionic balance from inside to out. This kind of transport work also requires your energy sources or your oxidation reduction potentials there. And also some cells also need mechanical work, where they of course are mortal and they need to move. So mechanical work would be in terms of motility in the flagellate bacteria or the ones that can glide or so. But within the cell also, structures are moved within the cell also. That is considered to be mechanical work. So cells have to do a lot of work. You may think of it as they are sitting around doing nothing, but they have to do a lot of chemical work, transport work, as well as mechanical work. They are not, just inert little cells that are sitting around doing nothing here. Okay, so we talk about microbial metabolism, and we are looking at, since we have looked at the 5 major nutritional types, a lot of this recycling into the nature, and we are just showing the nitrogen cycle here. Within the nitrogen cycle, we can see that when the nitrates to nitrites to ammonia to nitrites, and also through nitrates to ammonia through the nitrogen pathway, you can see a lot of these pathways are microbe dependent. Do you see that? So these microbes are actually running the whole nitrogen cycle for us. Just an example. You could look at other cycles also in nature. So microbes are very important, and microbial metabolism is very important in terms of maintaining the equilibrium of these elements such as nitrogen, carbon, oxygen, hydrogen. So just to look at this one, you can see the nitrogen cycle is definitely very dependent on different microbes, and some of these changes can only occur, or these metabolic pathways are only present in bacteria. Okay, so just giving you the importance of the bacteria in terms of metabolism and how important it is. So let's talk a little bit about ATP. You've heard of the ATP and you have probably understood it very good. Just to recap for you, ATP is considered to be the energy currency of the cell, which will drive a lot of reactions for us. And it is adenosine triphosphate. So adenosine is of course your nucleoside. The nucleoside always has the nitrogenous base and the sugar. We will call it a nucleoside. And when the phosphates are attached to it, we will call it a nucleotide. So ATP is a nucleotide, but adenine and ribose, this is nitrogenous base and a 5-carbon sugar ribose, they compose together as a nucleoside. Please remember the terminology. Now the nucleotide, which has a phosphate, can be an AMP, ADP, or ATP. So if one phosphate is attached, it's AMP. If it is two phosphates, it's ATP. And then it is ATP has three phosphates, right? So please really realize that this is the source of energy, and energy is usually by breaking this from ATP to ADP, you will be releasing energy. If you are making it from ADP to ATP, you're trapping energy into it. So this is your currency that will be used quite often when we look at the metabolic pathways. So what is the role of ATP in metabolism? The reactions are categorized as exergonic or endergonic. Exergonic is when you break down the ATP, coupling of the ATP, and give energy to another reaction. This would be an exergonic reaction, breakdown to produce energy. Endergonic is when you take away and you produce. So these exergonic endergonic reactions are tied together, where ATP or GTP will be providing the energy to run some of the reactions. So here is an endergonic reaction alone being shown, where A plus B has to be converted to the product C plus D. And you can see by the arrow that the forward reaction is stunted. It does not occur very easily. Okay, so if the endergonic reaction was to occur by itself, it would be very, very slow, and it would be a lifetime waiting for these products to be released. Was if it is coupled with the ATP breakdown, you can see your reaction goes much faster forward, and therefore you can make these products very easily within the cell. So ATP role of ATP in metabolism is very, very important, because it channels or it fuels some of these endergonic reactions which would not occur very easily. So my question is, what is ATP about? What is in that ATP? Because it is capable of transferring a phosphate to some other substrates, and this phosphate transfer allows the reactions to proceed also. So we talk about the group transfer potential of some compounds. Now you know ATPs, in your head, ATP is the one that will give you the maximum amount of, it has the highest electron phosphoryl group transfer potential as plus 35. But there are other compounds in the cell that we will look at now, which will be used in this. For example, your PEP compound that you will come across during glycolysis has a phosphoryl transfer potential of plus 62. That means it is a better phosphate donor than your ATP. So sometimes PEP will be used to drive reactions. There is another one which we will come across, is the phosphoglyceraldehyde, glyceric acid 131, which one has a plus 49 phosphoryl transfer group transfer potential. Your acetyl P again has a higher than your ATP. Glucose 6-phosphate has a lower phosphoryl transfer potential than your ATP hydrolysis. So I'm giving you examples of some of these chemical compounds that will drive reactions rather than just ATP. So as such, group transfer potential is the negative of the free energy hydrolysis, which is always denoted by delta G prime. So this is the potential which is always given in the positive way. So these are convenient ways of estimating the direction in which the cell will proceed. Okay? So here is a very clearly as to how the transfer reaction using the nucleophilic displacement will transfer a phosphoryl group from one of these compounds into another substrate. I think I have to stop now. Please study on in this chapter and think about it. The pathway is very simple, but all the nitty-gritty behind it is very important in studying for this chapter. So please review your glycolysis, and I will fit in all these ideas into your glycolytic pathway. So please sit down and make a glycolysis pathway for yourself before you come to class on Thursday. We'll stop at this, and if you have questions, I will wait. If not, Then have a good afternoon and evening.
Dr. Una,
Yes.
for the summary, for instance, part of the red group, so I write my summary on the assigned article for the red group, and then my questions are for the red group, or are they for the blue group?
For the blue group. That makes you read the other paper, otherwise
Yes,
you won't read it. Do you understand why my logic is that way? You are supposed to summarize one paper and you understand it very well. But the other paper, the other group are reading, you should be reading it and challenge them with questions then.
Thank you so much.
Presentation. Okay, good.
Yeah, just want to double check. Thank you.
Now, asking questions is very good. This sometimes when you ask in the class, I can explain it to others at the same time and they will get the idea also. But if you ask these questions in an email, you will the only one who will benefit for those question answers, right? So please try and always focus on questioning here when the group is still around. Thank you.
Have a great day, everyone.
Take care. Hands up. Bye.